All right, in this video, what we're going to look at is transport across membranes. It's the second section of the cell biology course for National 5 Biology. And as I said before, GCSE, National 5, this is the specific way it is taught in National 5, but it doesn't mean it's not relevant to other biology studies in other places because it's the same across the world. Biology is constant. Okay, so what we're going to look at first is the structure of the cell membrane, and we'll have a quick look at this here. So the main structure is a double layer of phospholipid molecules. So the phospholipid molecule over here, we have what we call the hydrophilic head and the hydrophobic tail, and they get together in what is known as the phospholipid bilayer. Now that's a pure phospholipid bilayer. Now the cell membrane itself isn't a pure membrane, pure phospholipids. In the cell membrane, there are lots and lots of proteins, integral proteins, that are involved in making sure that the membrane has a function. But remember that the hydrophilic heads are pointing towards either the exterior of the cell or the interior of the cell, where there are aqueous solutions, watery solutions, and the interior of the membrane is hydrophobic. So anything out here that dissolves in an aqueous solution is unlikely to dissolve in a hydrophobic solution, the hydrophobic area in the middle. So it prevents things crossing the membrane easily. Some things do cross the membrane, oxygen, carbon dioxide, quite freely, but other larger molecules find it very difficult. Okay, so we call the membrane selectively permeable, and selectively permeable is because the cell itself can select which proteins it puts into that membrane. So each cell determines which proteins are present in its membrane, and that's determined by a number of factors, transcription factors and things like that, that tell the cell what it has to become, what it has to make. But what it means is that every cell is different depending on its cell type. And that means that in order to determine what can cross a cell membrane, you need to know the cell type and you need to know what proteins are in the membrane. So for example, in liver cells, in a lot of liver cells, there are aquaporins. These are proteins that just have channels in them that allow water to pass freely, and they can stop the flow of water if they need to. So you get things like channel proteins. We also get transport proteins that use energy to transport things across. We'll talk about that in just a second. But remember, it's selectively permeable. Right, now, energy is not required for the movement of many types of particles because of the physical processes of diffusion. And diffusion has two forms in biology. You've got diffusion where substances that are dissolved in water diffuse from one place to another. And there's no membrane required for this in general. So if something is concentrated in one area, it will gradually spread through an aqueous solution so that the concentration of all the particles is equal across the whole volume of the space. And then we have osmosis. Now, osmosis is specifically the movement of water. Now, water will not move easily from one place to another against the flow of diffusion. Substances would have to be, the substances dissolved in the water would find it much easier to move than the water itself. So in order for osmosis to be seen, you have to have a membrane that allows the transport of water, allows the movement of water, but stops the movement of the other particles. And so if we look here at the two types, we see diffusion here. We've just got a concentrated particles in one corner of the beaker here, and then they eventually spread out over the beaker. It can be quite a slow process. Osmosis, however, what's happening here is that we have a higher water concentration here and a lower water concentration here, i.e. there's less water in this side per volume because particles are using up some of that space than there is in this side, and the water moves across from one side to the other. And you will see this happen. Eventually, pressure of gravity will stop that flow happening, but you do see this this movement, and you would get this. This is exactly what you would see if you had this set up. So, and it's precisely what we see in plants. So plants bring water into their cells, but eventually the hydrostatic pressure of the cell wall stops any further movement of water into the cell once the cell becomes turgid. All right. So Both of these processes move the substance that is moving from a high concentration of it to low concentration of it. So here we've got high concentration of the dissolved particles move to low concentration of the dissolved particles and eventually spread out. In osmosis, which is basically just the diffusion of water. Osmosis is diffusion too, but it's specifically about the diffusion of water across a membrane. And on this side of the membrane, we have high water concentration because there's few particles dissolved in it. On this side of the membrane, we have low water concentration because there's lots of particles dissolved in it and the water moves across the membrane. Okay, hopefully you get that. It's a tricky concept and causes pupils a lot of problems every year. All right, so here's diffusion. Dissolved substances moved from high concentration to low concentration down the concentration gradient. So high to low. I can't really do that. High, am I appearing? High to low. Okay, and it's down a gradient. So think about diffusion and osmosis as being on a bicycle at the top of a mountain. All right, and there's Trixie, my dog, joining in the lesson. So the dissolved particles up here we've got in the extracellular space. We have lots of particles and nothing on the other side of the membrane. Then the intercellular space gradually fills up so that the concentration of those particles is equal on either side. So in this particular case, the particles are able to move across the membrane. All right, it's important for oxygen and carbon dioxide movement across cell membranes for respiration. Okay, here's a little diagram of diffusion and respiratory gases in a cell. And I use a little diagram like that. It takes about a minute to draw once you learn it. And it's a good way to just make sure you've got everything written down. I might do a video on just how to make that diagram in the future. Okay, osmosis is the movement of water. Water moves from high water concentration to low water concentration. Again, down the concentration gradient, like a bicycle. No energy required. It just happens, right? The only thing that has to be manufactured is a membrane in between that allows water to move. And that's the key. Everything else has to stay put either side of the membrane. It's very important in plants. Plants use it as a way of getting water into their roots and transporting water up through their cells and into their vacuoles in their cells too. Because plants themselves are hydrostatic organisms. That's their skeleton is made of water. Okay, it's also a major process in kidney function, which you might learn about in higher biology. Here's a root hair cell. And again, we see that outside the root hair cell, we have water molecules at high concentration because there's nothing dissolved in them. Whereas the plant manages in the vacuole of this root hair cell, it fills it up either with salt or sugar, some kind of solute in there to lower the water concentration, which means that the water will flow from high water concentration to low water concentration. And then again, the concentrations of the dissolved substances are manipulated by the plant to cause the water to move up the xylem in water transport in the plants. We'll talk about that in another lesson in the future. But again, here's a little revision diagram. Should take about a minute or a couple of minutes to sketch out. It's always handy to have a little whiteboard. Do your sketches when you get 5 minutes for your learning. Okay, doing them over and over again is how you learn them. That's the only way. All right. The rate of passive transport depends on the distance, the surface area of the absorbing membrane, and the concentration difference. So in these tissues where passive transport is important, the transport is happening over very small distances. For example, in the lungs, carbon dioxide and oxygen are diffusing across just 2 or 3 cell membranes with almost no cytoplasm in between. They've got high concentration versus low concentration on either side and a very, very high surface area. So all absorptive or transport surfaces have these characteristics. High concentration differences. Surface areas of the absorbing membrane are very large compared to the volume of the cells involved, and the distance is very short because these things, as you can see here, I dropped some potassium permanganate into this measuring cylinder, a few crystals down at the bottom, and it diffuses quite rapidly up the first centimeter or so at the bottom, but then it takes a very, very long time for that to spread out across the rest of the measuring cylinder. All right, so just a passive transport summary. No energy is required. It goes from high concentration to low concentration, moves downhill, and most important for respiratory gases and water. Remember that the movement of water is called osmosis. It is still diffusion, but it's the diffusion of water, specifically across a membrane, a selectively permeable membrane, or a water permeable membrane. It has to be water permeable. The water has to be able to get across it. Okay, so let's have a look at the effect of osmosis in cells. So in animal cells, water moving in can burst the cell because nothing is going to hold the cell together. The cell is just made of phospholipid molecules in its membrane. That's held together with some other molecules like cholesterol and stuff that toughen up the membrane a little bit. But in the end, if the water keeps moving in, the animal cell will burst. Okay. In plant cells, we get a different situation because they have a cell wall. The water moves in. This is part of the plant skeleton. And the water moves in, pushes against the cell wall. Eventually, the pressure from the cell wall pushes back against the osmotic pressure coming in. And the whole thing, the movement of water reaches equilibrium with the cell full of water. This is called turgor, or turgidity. The cell becomes turgid. And this, of course, allows the plant to stand upright. In a normal situation, well, when it hasn't got enough water, a plant will be kind of floppy, or flaccid is a word that you can use. Flaccid. Some people say flaccid, but flaccid's the correct pronunciation, or the original pronunciation's not even worth fighting about anymore. And of course, if the plant cell becomes very wilted and loses all its water, it becomes plasmolyzed. The cell membrane in plant cells is attached to other cells on the other side of the cell wall. It looks like it's attached to the cell wall, but it's actually attached to other cells on the other side of the cell wall. Those attachments remain in place, but you can observe the plant cell, its vacuoles shrinking horribly, and then the whole cell pulls away from the cell wall, leaving only those attachments to other cells on the other side of the cell wall in place. So it forms this kind of star-shaped structure. This is called plasmolysis, or plasmolyzed cell. You need to know these terms: plasmolyzed and turgid and turgidity. Not quite sure which is the preferred one. In animal cells, of course, if they lose water to a lower water concentration outside the cell, and water flows out, they will shrink, but the cells can survive that. Depends on whether there's a major effect on their pH or their salt concentration. It can kill the cells as well, but quite often they can recover from that. They cannot recover from being burst. Okay, so isotonic is what you really want for animal cells. Otherwise, the cells become damaged. So plasmolysis and turgidity, here we go. So we've got our isotonic solution. There's equal amount of water coming in and going out. In the plasmolyzed cell, the water is leaving because the water concentration is lower outside the cell than it is inside the cell. So if you put plant cells into a very salty solution, and you may have done this for your assignment practical, the water leaves the cells lose water, they become plasmolyzed, and the whole tissue becomes lighter in mass because the water is moved out into the solution. So it's one of the classic experiments for osmosis in biology. And then if you put very low concentration water, here's me getting mixed up, very high concentration water outside the cell, the water will move from high to low, so it's going to move into the cell, and it will fill up the cell with water until it can flow in no longer, and the cell becomes turgid. So high water concentration outside the cell, low water concentration inside the cell, downhill. Okay, nice and simple. Although it's very, very, very easy to get confused. So it's really important to have your thinking cap on when you're doing these questions, and maybe sometimes to just draw out the situation. I've got a dog right beside me chasing her Kong. Yes, now she's annoying me. Okay, the last thing we're going to talk about is what happens if you want to transport stuff against the gradient. You're trying to push stuff uphill rather than downhill. Well, in that case, you need energy. And in National 5 learning, the only thing we have to remember is that to push substances from low concentration to high concentration, and this includes water, I guess, you need to have some method of putting energy into the process, and you have to have something that can do the job. And the job is done by a protein. The proteins are the machines of the cells, and those machines require energy in the form of ATP in order to make them work. So there's a couple of examples here. And so this is a sodium pump and a potassium pump, and what happens is that the ATP is used, turns into ADP. We're going to talk about that when we talk about respiration. And the little orange fellows here are the sodium, they get pushed out, and the little yellow fellows here are the potassium, they get pulled in. But it's not an example that you need to remember. The only thing we really need to remember about active transport is that it transports substances against the concentration gradient, pushes them uphill. You need a car to get uphill. You can use a bicycle to get downhill, but you need a car to get uphill, and you use energy in the form of ATP. Okay, and then we'll talk about ATP in our next lesson. So, oh, I haven't got my, let's see this. I think we'll stop there. Thanks for watching. See you in the next lesson.